Okay, so let's discuss just a couple of generalities about kidney function altogether. So by now you're aware that the major function of the kidneys is to filter the blood so that the blood plasma that's left over that comes out of the kidneys will have the exact concentration, content, and volume that you want, leaving behind what you don't want in a filtrate that eventually becomes the urine. So blood that is not yet filtered will enter the kidney through the renal artery, and then this unfiltered blood, even though it's not shown here in this drawing right here, this unfiltered blood will circulate throughout the entire structure of the kidney shown there, leaving behind the filtrate that becomes the urine that is going to occupy these areas right here. And then once that filtrate is produced, it will go through the renal calyx right there into the renal pelvis and then down to the bladder through the ureter. So that unfiltered blood is going to circulate all throughout the kidney tissue there, and this brings up a very important point. There are two major areas of tissue that you're going to find in the kidneys. There is a renal cortex and there is a renal medulla. And what we're going to find here is that the function of these two different regions of tissue is going to be very different. And the major difference is that the renal cortex and the renal medulla are going to have very different osmolarities, which is very seriously going to impact how we reabsorb water in those areas. So to clean up the last of kidney structure here, you're going to notice that the kidney itself has 8 to 15 regions that look like this. Those are called renal pyramids, and they make up the bulk of the renal medulla. So these renal pyramids are going to be where we find the nephron, which is the major, most basic unit of kidney filtration. So if you can imagine again that you have all this unfiltered blood that is circulating throughout the kidney tissue there, as that blood is filtered by each individual nephron, we produce that urine filtrate that goes through the renal calyces into the renal pelvis and then eventually down to the bladder. So on top of what's shown here, you can imagine that each one of those renal pyramids has thousands of nephrons that continue to filter the blood coming in. Okay, so let's move on to the actual structure of the nephron here. So the place to start here is the afferent arteriole, which is coming in from the renal artery. So at this point in the afferent arteriole, we have not yet filtered any of the blood. This afferent arteriole is going to enter into a capillary structure called the glomerulus, and the glomerulus is tightly interacting with this region of the nephron right here, which is called Bowman's capsule. So this region right here at the interface between the glomerulus and Bowman's capsule, this is where our very first kidney function is going to occur, and this is called filtration. So we are going to produce an initial filtrate that is essentially going to have the same osmolarity as the blood that was filtered, meaning that we haven't really done a whole lot to alter the content of things like water and salts. Now that this blood that has been filtered, it will exit through the efferent arteriole, and then what you're going to notice is that this efferent arteriole isn't just going to go off on its own, it's going to continue to intertwine with the entire length of the nephron until we get past the distal convoluted tubule here, in which case it then becomes the renal vein, which is going to allow that blood to exit the kidney right here. Okay, so let's just take this step by step as filtrate flows throughout the length of the nephron. So we said that our initial filtrate is formed right here in Bowman's capsule between the interface between the glomerulus and the Bowman's capsule membrane. This filtrate is going to be pushed throughout the entire length of the nephron. This first region right here, which is found in the renal cortex, anything above this purple line right here is the cortex, and anything below it is the medulla. We have the proximal convoluted tubule here, in which case we are going to reabsorb about 65% of the water that we produced right here. This filtrate is still going to have the same osmolarity as what it had previously, so it's still going to be isoosmotic to the blood plasma until this filtrate passes below the break line between the cortex and the medulla and enters into the descending limb of the loop of Henle. Once you get into the medulla, the osmolarity is going to change. The osmolarity of the interstitial fluid here is not going to be 300 milliosmoles like it was up here. It's actually going to be somewhere between 1200 and 1400. So the interstitial fluid here is going to be hyperosmotic to the filtrate that comes in. So because of the overwhelming number of aquaporin channels that we have right here, water will be able to flow from low osmolarity inside the filtrate to high osmolarity out here. So we're going to reabsorb a lot of water here as well. But as this filtrate makes its way down, we are going to increase the osmolarity of the filtrate because we are removing water without removing any solutes. We'll get down here to the vasorecta area, and then once we make it up into the ascending limb of the loop of Henle, this region right here in the thick segment is impermeable to water, but is very permeable to salts like sodium and chloride. So we will reabsorb a lot of sodium and chloride there to reduce the osmolarity of that filtrate even more, so that once we come back up here into the cortex, we are basically isoosmotic once again. But in fact, we're actually going to get hypoosmotic because the osmolarity up here is going to get as low as 2 or 100. Okay, so next we get back up into the cortex, into the distal convoluted tubule. So now we're going to see that we get further water reabsorption here, but most of the water reabsorption that we have yet to perform is actually going to happen here in the collecting duct, which will eventually take the mature urine down to the ureter. So there are a lot of aquaporin channels in the collecting duct, but what we have seen, especially in the case of antidiuretic hormone, is that we can adjust our rate of water reabsorption in the collecting duct based on the osmolarity in our blood. If the osmolarity of our blood is too high, then antidiuretic hormone will cause even more aquaporin channels to show up in the membrane of the collecting duct, so that we reabsorb extra water so that we bring the osmolarity of the blood plasma back down. So the whole point with the collecting duct here is that we can adjust how much water we reabsorb based on the needs of our body, especially as it pertains to electrolyte balance and osmolarity in general.